Jai Gurudev students. Today I will discuss with you that is the growth regulator or the phytohormones. Presence of a growth regulating chemicals in plants was first suggested by Sach in later half of the 19th century. The plant hormones acts as a messenger substance for growth regulation, and these hormones are organic substance which is required in small quantities at a low concentration, and their site of actions and synthesis are usually different. So today I will discuss about these growth regulating substances. Number one is auxin. These auxin are one of the most important, that is group of plant hormones. Auxin were first to be identified as a growth hormone, and IAA, that is indole acetic acid, is the principal naturally occurring hormone. The compound related to stop the activity of auxin are known as antioxins, and the auxins which easily can be extracted from the plant is known as free auxin. Few other auxins are there which are that is indole butyric acid, indole 3 pyruvic acid, phenol acetic acid, indole butyric acid, or naphthalene acetic acid, which is NAA, 2,4-dichlorophenoxyacetic acid. The function of indole acetic acid are of, that is, cell division, cell elongation, root formation, apical dominance, and prevention of abscission layer. This abscission layer means a layer which is forms in between the part of the that is plant and the main part of the plant, and due to which senescence takes place, and that part is get detached from the main part of the plant body. Now regarding the history of the auxin, so already I have discussed that is what is known as antioxin, then free auxin, and some auxin which cannot you that is extract from the plant easily, that is known as bound auxin. Now the history of auxin. So one of the history is 1880 by work of the Darwin, that is he found that in case of Coleoptile and Coleorhiza, I think you can remember the part. Coleoptile is a future shoot and Coleorhiza is a future root. Now this Coleoptile future shoot, one part is lighted, which is illuminated side, and another part, opposite side, is a dark part. And it has been found that when light it falls one part of the Coleoptile, then bending towards light takes place. Okay, curvature or the bending takes place. And again, if the tip of the Coleoptile is removed from the that is plant, then it has been found that that type of curvature is not found if light is fall or illuminated part is there also in the plant. And in another experiment, it has been found that if the tip of the Coleoptile is covered by the foil, then again the curvature will not form in presence of light also. So this one is the work of Darwin. Another work of Bason and Jensen, and he at that time he found that the Coleoptile bend towards light, okay, and if one gelatin sheet is inserted in between, that is the tip and the rest part of the Coleoptile, then also that is bending takes place. Now the growth takes place if a mica sheet is inserted in the lighted part, then also the curvature formed. But if that mica sheet is inserted into the dark part, that is opposite side of the illuminated part, then the bending is not occur. So from these experiments, they came to the conclusion that there must be some chemicals are there which get stimulated due to presence of the light, and that's why the curvature is formed. And when that tip is removed, then that chemical removed along with the axial part of the coleoptile. Okay, and here from this experiment it is clear that this light part is not that much of important than the dark part, means that chemicals move to the dark part and cell elongation takes place in comparison to the light part. That's why it moves towards light. But when the dark part is interrupted by the mica sheet, that curvature is not formed. So another work of the Pearl, and he found that if the decapitate means remove tip, it just turn over, then also that is curvature, that is the growth takes place. These are the history. Now the isolation and the molecular structure. In 1931, Coghlan and Thyman in 1935 isolated auxin and studied their chemical nature, and the isolating auxin form urine of a pathological Dutch patient, and which is identified as a auxin B. Few other auxin are present. Already I mentioned the name. So auxin present generally in case of the plant in the meristematic portion, and in case of the human beings in urine, and particularly in the person suffering from a disease from pellagra. But role of the indole acetic acid in human is not clearly understood yet. So this is the structure, chemical structure of indole acetic 3 acetic acid, indole 3 butyric acid. I have given you only few of the structure. Now the uses of the auxin for the eradication of the wheat, because high concentration of auxin like the 2,4-D or 2,4,5-T. This D is dichlorophenoxyacetic acid. This one is dichloro. So, and that is overstimulated the growth promoting activities of the cell of the root. As a result, the root get distorted and the seed tube get blocked, and that's why these are two used as a weedicide. Okay. Now next one is root initiator or root initiation. Auxin like IAA, IBA, NAA, these are in a low concentration form 10 to 1000 parts per millions on the cutting initiate adventitious root formation. Flowering initiation, again 2,4-D and NAA, these two auxin helps for the formation of flower. Now next one is productions of parthenocarpic food, seedless food due to the presence of auxin, application of auxin, apical dominance and dormancy. Shoot growth in nursery trees and bushes can be reduced, particularly during storage and transport. Next one is enzyme activity. Auxin that stimulates the activity of enzyme, and they show inhibitory effect on isoenzymes like the peroxidase in tobacco. To prevent the premature fruit drop, it prevents the formation of abscission layer. Now, one of the bioassay. Bioassay means detection of a plant growth substance using a biological test material is called bioassay. How to synthesize? So this one is these are the tip of the coleoptile within a agar. That one is placed, and after that these are removed and these agar blocks are cut up into pieces. That piece is placed on a, that is coleoptile, and where the tip part of the coleoptile is removed, but still then it will show the curvature. Means the chemical substance that is present within this that is tip of the coleoptile, it will come to the, it absorbed by the agar, and that agar when placed on the cut tip of the coleoptile, that chemicals pass to the coleoptile, due to which curvature is formed. So this one is one of the bioassay.